First of all, we'd like to welcome all our guests here in person and those watching over the online webcast. Welcome to our 2016 CCOVI Lecture Series. Today the tables have been turned somewhat, and I have the pleasure of introducing our esteemed leader, Dr. Debbie Inglis. In addition to leading CCOVI, Debbie is one of the Institute's core scientists, as well as an associate professor in the Department of Biology at Brock University. She received both her BSc and her PhD in biochemistry from McMaster's University in Hamilton and has been involved with the grape and wine industry her entire life. She's also a Niagara-based grape grower, so she understands all aspects of it. She's led multi-partner research initiatives funded through industry, the province of Ontario and the government of Canada on industry priorities. Her research has focused on topics such as grapevine cold hardiness, removing lady beetle taint from grape juice, Appassimento and sparkling wine production, as well as identifying organisms and control strategies for grape sour rot. She also has an established NSERC-funded research program on ice wine with a focus on wine yeast biochemistry and improving wine quality. Today she will be presenting to everybody developing Ontario Appassimento wines, the impact of drying method, yeast strain and botrytis on wine quality and consumer choice. So please welcome me in welcoming Debbie Inglis.
Thank you, Belinda, for that lovely introduction. So I'm here to talk about a very large project that we've been doing for the past 5 years in partnership with industry and partners at Vineland Research and Innovation Centre and Niagara College. So a few years ago, we got together with the industry, and if you're from the region, you know that our weather and climate the last few years is really starting to become a little more volatile. We have extreme years where it's very hot and dry. We have other years where it's very wet and cool during the growing season. And the industry wanted to start to look at techniques that would help mitigate these fluctuations in the climate and still allow them to produce high-quality red wines consistently year after year. So we wanted to look at techniques where we could further develop flavors in the fruit given all of these consequences of our climate during the growing season. So in looking around at what's happening in other parts of the world, we wanted to start to look at technologies that were already in existence that we could look to adapt here for Ontario. So in looking at that, there was a technique in northern Italy where the grapes are harvested off the vine at regular harvest time and then further ripened off vine in barns, and this is known as the Appassimento method. And the technique was developed because in that region they weren't able to get the full flavor development of the fruit during their growing season. So we thought, you know what, this is a technology that we think may have really great application here in our region in Ontario. And so with that, we developed a large program to be able to assess this technology. Now, in terms of producing Appassimento wines, there are a few key factors that we needed to keep in mind as we were developing the program. One is that there can be an increase in oxidation compounds that build up in the grapes during the drying process that can then get translated into the wines. And so we wanted to be cognizant of that, making sure that we were looking out for some of these oxidation compounds, which would be compounds like acetic acid, acetaldehyde, ethyl acetate, and making sure that we were measuring them through the drying process as well as in the final wines. In addition to that, there's a lot of unknowns at this point in terms of whether there's some benefit or no benefit at all to having some botrytis infection in the fruit that occurs during the drying process. Botrytis can build up in the fruit given the right humidity levels and temperature in drying conditions. So we wanted to again be cognizant of that and looking for the impact of that in the project. So we split the project up into 3 different questions. And the first one, we wanted to compare 5 different drying techniques that could be used for further ripening the fruit. The second was a yeast strain trial where we wanted to look at the ability of a locally isolated yeast from the skin of Riesling grapes, and that was known already to us to be a low producer of oxidation compounds. And might it have application in this Appassimento style wine production? And the third question we wanted to address was the impact of botrytis on the fruit. And whether there would be any impact on the consumer preference of the resulting wines with and without botrytis developing on that fruit. So I'm going to walk you through all 3 projects. There's a lot of information here, but it's very exciting and I think is going to have a really tremendous impact for moving our industry forward using this technique. So the 5 different techniques that we were using for drying the fruit, we started with our control, where we would just hang the fruit on the vine and let it continue to further ripen during the fall season. Okay. We then also took a look at traditional barn drying, and this was from our partner at Cave Spring Cellars. We investigated a specialized drying chamber that was developed at Vineland Research and Innovation Center, where the temperature and humidity could be clearly controlled, very tightly controlled. We wanted to look at utilizing greenhouses. At the time that grapes are ready for harvest here in the region, there's some open capacity in greenhouses in the floriculture industry. So we thought, gosh, could we make use of that open capacity for a few weeks to dry the grapes? And the last technique that we looked at, or the last drying condition, was using refurbished tobacco kilns. So this kiln setup was from our partner at Rife Estates Winery. So with that, for each of these drying techniques, we wanted to really make sure we were following the environmental conditions. We wanted to monitor the temperature and humidity throughout that drying process to really understand what was going to cause some of the differences that we were hypothesizing would result in the fruit and in the final wine. So we actually harvested and dried grapes over 4 different consecutive seasons. We used Cabernet Franc grapes for the various drying regimes. The first year, it was a smaller study, and we were basically just working out all of our methodology. And then once we were clear on how we were going to run this program, the following 3 years, we compared all of those techniques. And we're just in our final year right now, and we're finishing up on some chemical analysis of the volatile aroma compounds and really bringing all of that data into the fold to understand the differences that we see sensorially and chemically in the wines. Now, all of the grapes for this project were donated from Pilotary Estates Wines, and the on-vine drying, of course, was done at one of their vineyards with the fruit just left on the vine, whereas the remainder of it was harvested for the various drying techniques. Our target Brix at harvest were 23 Brix. So this fruit, the Cabernet Franc, it was ripe. It wasn't underripe fruit. It wasn't overcrop fruit. It was ripe fruit that we were targeting 23 Brix before harvest. Through the drying techniques, we were bringing them up to 26 Brix and 28 Brix. And then all of the wines that were generated from the fruit were fermented in triplicate. And using the same protocol, and the yeast we use for all of these trials is an industry standard EC1118. And that is the standard yeast that's recommended and typically used in northern Italy to make Amarone wine. So it was recommended to us by Laumont. So I'm going to go through some of the temperature and humidity data across the 3 years for the various drying techniques. So as you can see from this diagram, the conditions were quite variable out in the vineyard. So you can see lots of variability in the temperature, day highs and lows. As we continued through the season, we got some days where the temperature was below 0 degrees Celsius, and lots of variability in the humidity as well. The humidity is in the blue lines. And we saw similar trends in 2012 and also in 2013, but I'll draw your attention to 2013 because you'll see quite a few more days where the temperature was low and stayed down well below 0 degrees, and on one day we almost hit minus 15, minus 18 degrees Celsius there. So these grapes, if you're going to dry them on the vine, they're being exposed to a lot of different conditions and risk out in the vineyard. So the fruit is exposed to climate risks. There's rain, fog, dew, wind, freeze-thaw cycles, and of course, there's wildlife that can look at these grapes as something nice to eat through the fall and early winter months. So lots of variability in temperature and humidity. When we look at the conditions in the barn, we also see quite a bit of volatility in the humidity and the temperature. It's not quite as variable as what we saw in the external conditions out in the vineyard, but these conditions in the barn correlated very highly with what we saw out in the external environment. And for our barn condition, there were no other inputs into the barn in terms of heating to change the internal environment. There were some fans to help circulate the air, but that was it. The benefit in the barn, though, is that the fruit is protected from the rain, it's protected from the wildlife, so it's not as highly impacted by those external climatic conditions. The greenhouse, we had a little more control over the temperature in the greenhouse. So in this first year, when the temperatures started to get a little too cold for where we wanted the temperatures in the greenhouse, we were able to apply some heat to bring that temperature back up. So again, with the greenhouse situation, a little more control over those internal environmental conditions, but the one element that was not as easy to control as the humidity level in the greenhouse. So when the heat was turned on, we were able to keep the humidity down, but in 2013, we did have quite a few days where the humidity was getting up above 90% relative humidity. And I draw your attention to that 90% relative humidity because when the humidity gets above that, that's when botrytis can really start to build up in the fruit. The kiln, just to put into perspective for you, although I've stretched the graph out, the kiln drying occurred over a very short period of time. It was really only a number of days to reach the 26 and the 28 Brix. We were targeting the temperature to be around 30 degrees Celsius. There was lots of airflow in the kiln. Of course, the fruit's protected from the rain and from the wildlife. And when we got the temperature turned on, the humidity would drop. When we'd open up the door and take the fruit out, the temperature would drop, the humidity would build up, we'd turn the temperature back on, the humidity would drop. So again, much shorter period of time to dry the fruit to get to the required Brix at this warmer temperature. Now, in 2012, we had a little bit of a mishap. Sometimes this happens when you're doing experiments out in a commercial setting. Somebody at the winery, for some reason, thought that we wanted the temperature at 40 degrees and not 30 degrees, and thought they were doing us a favor and turned the temperature up to 40 degrees. So the first day, the temperature was up at 40 degrees, and the fruit only took one day to get to 26 Brix. Then we were able to dial it into 30 degrees. The following year, again, a bit of a mishap with the kiln, and we had some problems with the propane heating system. So we had a couple of days where we just weren't able to get any heat in there. So the drying took an extra day to reach the desired level. And the last condition that we're looking at is the drying chamber, and you'll see a very different look to these graphs than the previous graphs. Very tight regulation of the temperature in red and the humidity in blue. And the first year that this experiment was run, the temperature started out at 10 degrees, but then they made a decision to drop the temperature down to closer to 5 degrees Celsius for the rest of the drying. And they did this because they, for this particular drying treatment, we wanted to try to stretch out the amount of time for the drying for the grapes to reach the Brix. And the humidity was kept very tightly around 80% relative humidity. The following year, the humidity started out a little bit higher. Temperature was still fairly low, just above 5 degrees, and then dropped a little bit later as the humidity was dialed in to drop lower. I will say in 2013 there was a little more botrytis on this fruit that developed, and I'm surmising that it's because of the early days where the humidity was a little bit higher. So I think what you can see from all of these conditions is that, yes, when the fruit is harvested and put into these various drying treatments, they are protected from the external elements, but each drying procedure has its own challenges to overcome, has their own benefits as well, but we were really interested to see what would happen with the fruit and what would result in the wine. So again, just schematically to illustrate for you the amount of time required to dry the fruit from each of the treatments. Here on this scale, the Brix are going up to 28 Brix. This was our target. And on the bottom scale for each year is just the amount of time required to reach that. And you can see in each of these years, the purple line indicates the kiln. And the kiln was really only taking a few days to reach our desired Brix level. For on vine and for the drying chamber, they tended to have the longest period of time required to reach the desired Brix. For the drying chamber, that was by design. And in the last year, 2013, that we tested the drying chamber, the actual drying time, the number of days to get to the 28 Brix was 88 days. It was our longest drying treatment. But again, for the drying chamber, that can be, the amount of drying time required can be controlled by the temperature and humidity in that environment. So with the experiment, we've got a great set of different circumstances to dry the fruit, from a very short time with the kiln. Intermediate drying times came with the barn and the greenhouse, and the longest drying times were coming with the drying chamber. So again, just to remind you, things to watch for in Appassimento production, the development of some of these oxidation compounds. So what happened with these compounds? What we found really across the board for all the drying treatments, we did not see very high levels of acetic acid that developed in any of the treatments across any of the years. But one treatment did stand out, and that was the kiln. So each year, although these levels on a relative scale are not very high, it did produce the highest amount of acetic acid in the fruit during just those few days of going into the kiln and the fruit being dried. Now, with that being said, in 2012, the level that developed in the kiln fruit was still low. It was still only 0.13 grams per liter. And to put that into perspective for everybody, when grapes are delivered to wineries at harvest time, the wineries start to get concerned when the acetic acid is at that 0.2 to 0.24 grams per liter. We certainly weren't close to that in any of the years. All of the other treatments, though, kept the acetic acid values quite low. We did try to see if there was any correlation with acetic acid production with acetic acid bacteria on the fruit in the kiln, and we just did not see that correlation. So we're still not quite understanding where the acetic acid was coming from. It could have been from another organism. It could have been internally in the fruit. Acetaldehyde also increased for all of the treatments in each year. but again, the values did not get very high. They came up to about 12 milligrams per liter, and again, that's fairly low for a level in the fruit. The malic acid did drop in all the treatments. So keeping in mind that the fruit is concentrating and the malic acid was either staying the same in concentration or going down in concentration, it meant that some of that malic acid was getting degraded during that drying treatment. And typically the malic acid in the final fruit was between 2 to 2.5 grams per liter. Glycerol did tend to increase in all of the drying treatments, and I'm just going to show those graphs quickly. But again, the levels would range typically between 1.5 up to 2.5 grams per liter in the fruit in 2011. 2012, the fruit was so clean, there was absolutely no botrytis. We actually tried to do some botrytis experiments that year, and there was just no inoculum on the fruit. And corresponding with that, we saw very low glycerol levels in the fruit. And glycerol is a metabolite that botrytis can produce when it grows on the fruit. In 2013, we were back up to levels between 1 and 1.5. So again, although some glycerol being produced likely from botrytis on the fruit, not at very high levels. So we also evaluated a lot of other chemical compounds in the fruit during the drying process. And my colleague Vincenzo Deluca is in the audience today, and this is some of the research out of his lab that was part of the program. And they really wanted to evaluate the polyphenolics that developed in the fruit, and were there differences among the different drying techniques. There's a lot of data here, and I wouldn't be able to do it justice by just explaining it in a few slides. I've got just some summary slides here, but we'll have to evaluate this data in much more extensive way compared to all of the other chemical and sensory data that we're collecting on these wines. But they evaluated between 25 and 30 different polyphenolics and how they changed in concentration during the drying, and grouped them into all of these various categories. So any of you that are interested in polyphenolics, like Belinda, you're probably having a heyday here. But this is a tremendous amount of work that was done over the 3 years for each of the drying treatments, for the fruit going up to 26 Brix and for the fruit going up to 28 Brix. To summarize, if I can give the big picture of what was going on here, many of the polyphenolics did increase by 10 to 30% during that drying process. And they saw the increases in some of those simple phenolics that were on the previous graph, in resveratrols, procyanidins, flavonoids, and in the anthocyanins. The resveratrol levels tended to be higher in the fruit in the years where there was more disease pressure, and again, that makes sense because that's a normal response of the vine out in the vineyard. The metabolite concentrations achieved were not specific to the drying technique, and the polyphenolic metabolite profiles have not yet been compared to the sensory profiles of the wine. We just recently completed and compiled all of the sensory data for the wines, and what we would now like to be able to do is overlay some of the phenolic data to see if there are some trends, and similarly, when we finish with all the volatile aroma compound data, we'll be overlaying that on the sensory data. The focus now in Vincenzo's lab is now shifted to looking at some gene expression analysis in this fruit during the drying time, because one of the really interesting results from this study was that these berries, even though the clusters have been taken off the vine, they're still very metabolically active, and they're very metabolically active in changing the polyphenolic profiles in the fruit. So there's a lot more to the story that his lab group is continuing on with. So when we did that work looking at the polyphenolics, we weren't separating out the skins and the seeds. And one of the questions that came up by our industry partners is what happens to the seeds during the drying process? Are the seeds ripening? Are they hardening off during the different drying techniques? And are there differences between the drying techniques? So we initiated a project to actually start to look at that. And Belinda Kemp spearheaded this and started to separate out the seeds from all of the different treatments and took a picture of the seeds when we first harvested the grapes, when they reached 26 Brix, and when they reached 28 Brix. And this top line is showing what happens to the seeds when the fruit is left out on the vine in the natural environment. Then we did the same comparison for the seeds from the kiln-dried fruit and for the other treatments as well. What you're going to see is the seeds from the fruit left out on the vine. I hope you can see this with the lighting, but they do look a lot darker. The seeds have hardened off more, typically of what we would see as fruit continues to ripen out in the vineyard. But for the kiln-dried fruit, there were still a lot of green seeds. They didn't look so different from what the seeds were looking like at the time of harvest. Additionally, for the greenhouse, we saw maybe a few more riper seeds, but again, not so different from the starting seeds. Similar story for the barn, and also a similar story for the drying chamber. So, yes, we can see when the fruit hangs out in the vineyard, the seeds are ripening off, but none of the drying treatments were really allowing for that same hardening off of the seeds as what we saw out in the vineyard environment. Well, Belinda took it a step further and further analyzed the tannins that could be extracted from both the skins and from the seeds. So I'm going to start with the seeds. And when we take a look at the on-vine seeds, so these were the seeds that were that much darker when they reached 26 and 28 Brix. We could see that there was an increase in the tannins that were being extracted from these seeds. This was a bit of a surprise to us initially because we thought as the seeds hardened off, we wouldn't be able to extract as much out of the seeds. But we've realized too that part of our extraction process is a little bit harsh on the seeds, and we're probably damaging the seeds somewhat through that extraction process and allowing more to be extracted than what typically would happen during wine fermentation. For the kiln, there was really no difference, no significant difference in the amount of tannins that were extracted from the seeds compared to when the seeds were first harvested from the fruit. However, for most of the other treatments, we were seeing an increase in the seed tannin levels. So they were, even though it didn't look like it from the pictures, they were behaving more like what the seeds were doing from the on-vine fruit. So we need to do a little more investigation here to understand what the impact of this would be during wine production, because of course when we're fermenting these wines, they're fermented on the skins and the seeds, and so we want to be cognizant of how many of these tannins we're extracting into the wine, because they can be bitter and astringent, and too much of it is extracted, it can have a negative impact on the profile of the wine. In terms of what was happening with the skins, for on-vine fruit, we actually found a drop in the skin tannin as the fruit was hanging out in the vineyard and reaching those higher and higher Brix levels. But this was not the same case for all four of the drying treatments. In the other four drying treatments, really we weren't seeing a drop in the tannin levels in the skin, and for the drying chamber, compared to what we started with, we actually saw a slight increase, significant increase in the skin tannins. So again, this will have implications during wine production. We actually would like an increase in the skin tannins in the fruit to be able to extract more into the wine during fermentation. All right. Before we get into the sensory evaluation of the wines, I just want to remind everybody that these are high alcohol wines. The fruit was dried up to 26 Brix and 28 Brix. And so the 26 Brix wines are typically ranging between 14 and 15% alcohol, and the 28 Brix wines are ranging between 15 and some up to 17% alcohol. So these are fairly high alcohol content wines, and that had to be taken into account in the experimental design to make sure that the individuals doing the descriptive analysis weren't getting palate fatigue from this very high content of alcohol. All right, so getting into some of the descriptive analysis. It's a complicated story, and I'm going to walk through some of the data here, but what we found is that there's not a consistent trend from year to year of what the profiles were and how the wines differed from each other given the different drying techniques. So there was vintage variation that we were still experiencing with these wines. But with that, I do want to walk through some of the results here. So if we start here in the first year for the on-vine fruit that was dried up to 26 Brix, we see that they were mainly described and separated from the other wines with higher descriptions of olive and green vegetal characteristics. Okay. Now, the drying chamber and the greenhouse wines were more similar to each other than the other 26 Brix wines, and they were described as more dried fruit and had more of a distinctive purple hue. Okay, so those two wines were a little more similar to each other in this year. The barn 26 fruit wines were described more with spice and a little bit more of a red hue to them. Okay, and the 26 kiln wines had a higher herbal impact and separated into a different quadrant in the PCA analysis. So what can I say about the 26 Brix wines? The drying treatment did impact the flavor profile of these wines, and it did separate them through this PCA analysis. Now, if we take a look at the wines that were then dried up to 28 Brix using those same drying techniques, we get a slightly different analysis of the wines. Now, the on-vine 28 Brix wine still had that same characteristic of olive and green vegetal. But then we start to see that the greenhouse 28 Brix wine shifted up here into being stronger in spice and more of that red hue that separated it out. The drying chamber wines were found to have were higher in astringency. Sorry, I can't read this very well. And bitterness and heat. The kiln wines were quite distinct from the other 28 Brix wines in that they had a little more of a brown hue to them. But you remember back in the environmental conditions, the temperatures got up above 35 Brix, or 35 degrees Celsius that year. So subsequent years, we tried to keep it around 30 degrees Celsius. So again, we're seeing that the wines are separating out. They are different and being described differently by the panelists. In 2012, what we found was quite a different scenario here. We were finding the 26 Brix wines more often clustered together in the descriptive analysis, so they had more of the same similar traits, and the 28 Brix wines clustered a little more together. And we found that similar type of result in 2013. So this was quite interesting for us to be able to denote for the different years. So the different drying techniques do give different attributes to the wine, but there is some vintage variation, and there's also variation if the fruit is dried up to 26 Brix versus if you're taking it all the way up to 28 Brix. It's a tool that winemakers have to try to funnel the type of wine and the descriptors that they would like to develop not the descriptors, but how the flavors in those wines. They have a choice of drying technique and up to what Brix they want to dry that fruit. Because I'm going to emphasize to everybody here, the wines were made originally from all the same fruit. Okay, all the same fruit came from the exact same vineyard, was harvested on the same day, and then was split into the different drying techniques. And from that, we resulted in all the different attributes in those wines. So there's still a lot more to this story on the volatile flavor compounds. We've gone through and measured all of these compounds for all of the wines for each year, 11 wines each year, done in triplicate and evaluated for all the volatile flavor and aroma compounds. We now need to compile all of that data and match it up with the sensory data. So we have a little more work ahead of us, Linda and I do to sort out that story. One thing we did want to touch on for the different drying techniques were a little bit of the economics. And we took a stab at this by trying to determine whether we would require an increase in grapes from the different drying techniques to result in the same volume of must compared to the control. Okay, so the control that was not dried at all, just the fresh fruit that was processed. So a certain amount of grapes were required to get a volume of juice for fermentation. We wanted to compare for all the different drying techniques how much more fruit would be required. And probably not really much to your surprise, the on-vine drying treatment required the most amount of fruit consistently year to year to be able to get the same relative amount of juice for fermentation. So out in the vineyard, yes, we have some degradation occurring of the fruit. We've got predation of wildlife, and all of that is impacting the overall final volume of fruit that we've got to process. The kiln consistently over the 3 years was the most economical in terms of not losing a lot of fruit. And again, I think that makes sense for everybody because the fruit was only in the kiln for a few days to reach the desired Brix. The barn and the drying chamber were quite similar to each other. And the other outlier was the greenhouse. But I have to qualify this for the greenhouse because we had an interesting scenario develop in the greenhouse, and there was a resident raccoon that we were having difficulty getting out of the greenhouse, and the raccoon loved the Appassimento grapes. And so the numbers here for the greenhouse are skewed because we lost fruit because of the raccoons. So it's not a fair comparison to bring the greenhouse into that analysis. All right, I did want to touch on the other two projects. So Project 2, we wanted to look at yeast choice that winemakers could have. And we had available to us a yeast that was isolated from the skin of Riesling ice wine grapes. And we had done some preliminary analysis with this yeast. It wasn't a strong enough fermenter for ice wine production up at 35, 38 Brix, but we thought maybe in Appassimento wines at that 27, 28 Brix range, it might prove to be very valuable. And why we were extremely interested in this yeast is because it's a very low producer of ethyl acetate and acetic acid. So we went through and developed a project for this, and this data that I'm going to present is from Jennifer Kelly's PhD thesis. She's a grad student in my lab. So the yeast that we're evaluating is a natural Saccharomyces bayanus yeast that was isolated from the skins of Riesling grapes from a local vineyard. So again, we were most interested in applying this to Appassimento wine production because of its tendency to produce low levels of oxidation compounds. So our concern is that through the drying process, on a commercial scale, if you do tend to increase these oxidation compounds in the fruit, you don't want to further increase them during the fermentation into wine. And so if you can use a yeast that's a low producer, you can keep those levels within a safer range in your finished product. So we really wanted to characterize this yeast and look at the upper sugar level that it would still be able to ferment and produce a high quality wine. And of course, in order to do that comparison, we wanted to compare it to the gold standard EC118 yeast that's typically used for Appassimento wine production. So we followed the fermentation kinetics, looked at the oxidative compounds in the finished wines, and Jen did all the sensory profiling through descriptive analysis of the wines. But we didn't end there, because at the end of the day, with this being a new yeast that potentially has some marketability, we wanted to see if there was some consumer preference for wines produced with this yeast over the gold standard EC118. So in the experimental design, we were comparing the fermentation of the wines with Saccharomyces cerevisiae compared to Saccharomyces bayanus, our isolate. And we used fruit that ranged from 21.5 Brix. All of the fruit was dried in the barn setting at Cave Springs, and we brought it up to 24.5, 26, and 27.5 Brix. And then we fermented each of these batches of wine in triplicate with either the cerevisiae yeast or the bayanus yeast. And from the fermentation kinetics, what I can tell you is that there really was not a big difference at all, nothing significant between EC118, Saccharomyces cerevisiae, and our bayanus yeast. In the control fermentations at the 21.5 Brix juice, at the 24.5 Brix, and at the 26 Brix. We started to see a little bit of a lag when we were up at the 27.5 Brix fruit for the bayanus. It took a couple more days to get to dryness compared to the EC118. It wasn't an extended time, but we did start to see a little bit of a lag there to get to dryness. In terms of the ethanol levels that the yeast produced, there was no significant difference for each of the Brix levels tested between the two yeasts. So they were taking in the sugar and converting it into ethanol, and no difference that was resulting. But where we did see the big differences were in those oxidation compounds. So for each of those Brix levels, we found that the EC118 made significantly higher acetic acid and ethyl acetate compared to what we found for the bayanus yeast. And this was very exciting for us. Same sort of results that we had seen in the preliminary ice wine fermentations. But at the end of the day, what really matters is what the wines taste and smell like in the glass and what the consumer acceptance is. So Jen carried out descriptive analysis on these wines. She trained 11 panelists to do this analysis over a number of months and used a 15 centimeter line scale in her analysis. So I'm first going to start by just showing you the wines fermented by Saccharomyces cerevisiae, and either at the 21.5 Brix initial juice, the 26 Brix juice, and the 27.5 Brix juice. And on this spider graph, all of the attributes that were evaluated in the wines are plotted out, and it's so that we can compare what the shape of the graph changes to when we're looking at Saccharomyces bayanus. The descriptors that are underlined are all the ones that were significantly different between these three treatments for the Saccharomyces cerevisiae. So probably not so surprising to you, as the Brix level went up in the fruit, there was more alcohol detected in the final wines. There was more astringency in the higher Brix wines, and there was more acidity in the higher Brix wines. In addition, they were higher in their earthy, toasty flavor attribute. They were more herbal in their descriptors, and they were less, at the higher Brix, less described with red fruit and candy confection. Okay? So that was for the Saccharomyces cerevisiae fermented wines. How does that change when we start to look at the Saccharomyces bayanus wines? Some things were similar. The higher Brix wines were higher in alcohol, okay? But they were significantly lower in acidity. And they were, significantly lower in acidity. They showed a higher tendency towards that earthy toastiness, and that was the same as the Saccharomyces cerevisiae. But they were also significantly higher in spice and black fruit attributes. Okay? Similar to the Saccharomyces cerevisiae, the higher Brix wines were lower in red fruit and lower in the candied confection. Okay, so if we now just focus on the high Brix wines for the bayanus and the cerevisiae and look at a PCA plot, you'll see something quite interesting occur here. The three different replicates of the bayanus wines completely separate out from the three replicates of the Saccharomyces cerevisiae wines. And again, we can see that the bayanus wines are higher in this black fruit flavor, higher in the earthy toasty flavor, whereas the cerevisiae wines are more astringent, more bitter, and higher in acidity. Okay, so this was quite striking for us to see with this bayanus yeast. So to summarize all of that data, we definitely saw that the Saccharomyces bayanus yeast shifted the profile of the wines towards more increased black fruit and aroma when we got into the Appassimento Brix levels. We felt that it has demonstrated some application in industry, but we wanted to continue on with some consumer preference work. But you have to wait for that result for just a few slides because I need to first talk about some botrytis work and then we'll go back to the consumer preference study. So the third part of the study, last part, is looking at the role of botrytis in producing these wines. So I can tell you in northern Italy, I was at a conference a few years ago, and they're still arguing over whether there should be a little bit of botrytis in the fruit to add complexity, no botrytis, or significantly more botrytis. And they seem to somewhat agree, and I say that loosely, around that 20% botrytis infection is still okay to add complexity to the wines without taking away from the quality of the wines. Well, we wanted to do some similar studies here with the fruit and look for our grape varieties, Cabernet Franc, that we were fermenting. Could we afford a little bit of botrytis infection in the fruit, and would it add some complexity to the wine? Or do we really need to be diligent and make sure the fruit is clean, and if any botrytis develops, we discard that fruit, get it out of the drying conditions so it doesn't further spread? There are some economic impacts here for producers of this style of wine because really most of the drying techniques other than the kiln produced a little bit of botrytis on the fruit during that drying process. So we decided that we would investigate the difference in the wines with 0% botrytis versus 10% of the weight of the berries infected with botrytis. And so for those of you not familiar with what botrytis looks like, I'm not sure if this will show up very clearly with the lighting here, but in uninfected berries, they're more of a deeper blue-black kind of color. The infected berries start to take on this reddish tinge when the botrytis is growing internally in the fruit. When it starts to sporulate, you'll start to see this gray fuzzy mold start to develop on the outside of the fruit. So we wanted to first separate the berries into the control black berries, the red berries, and sporulating berries, and make sure that what we thought was botrytis was really botrytis. So we separated out the fruit, and we did a few experiments. One was an experiment that Wendy McFadden-Smith, my colleague, helped us design, where we just put this fruit in a humid chamber and let it sit for 8 days and looked for the development of the spores on the surface of the berries. And what we found is that for all of the red berries, yes, they all developed the fungus. For most of the blackberries, they didn't. And for all of the berries that were already sporulating, they developed more of these spores. We crushed the fruit up. We further plated it out to make sure that what we thought was botrytis was really botrytis, and definitely it was botrytis. You can also see, if we take a look at the color of the fruit, the red berries were browner in the must color, and the sporulating similar. Definitely not the bright red color of the blackberries. We also saw in the fruit that there was an increase in glycerol in the red berries and the sporulating berries, and an increase in gluconic acid over the control. And again, these are typical indicators, metabolites that botrytis produces when it's growing. So then we were off to work. The lab group meticulously went through the fruit and separated out the botrytis berries from the clean berries. And we ended up with batches of fruit that was completely clean of these botrytis berries, and berries that had that red tinge to them that would contain the botrytis. And then we blended back in up to 10% of the weight of these botrytis berries. So we had a fermentation with completely clean fruit and a fermentation that had 10% of the botrytis infected fruit. And when we took a look first at just the juice analysis, really everything that we measured, Brix, pH, TA, acetic acid, glucose, fructose, there was really no difference between the clean fruit and the fruit with 10% infection. But there was a slightly higher level of glycerol and gluconic acid, as expected, in the fruit that had 10% botrytis present. We carried out the fermentations. There was no difference in the fermentation kinetics between the two conditions. We looked at the final wines, and for pH, TA, ethanol, residual sugar, acetic acid, and acetaldehyde, there was no difference in the wines, and just a very slight increase in glycerol and gluconic acid in the wines that had the botrytis infection in them. When we carried out the sensory analysis, we were very pleased to see that really there were no significant differences in almost all the attributes that were evaluated except for dried fruit aroma, and it showed a bit higher intensity for the wines that had the botrytis infection in it. But all the other attributes, other than color, we couldn't evaluate color because it would have let the individuals know that there was a difference in the wine. All of the other attributes, there was no significant difference between the wines. So now we got on to the consumer preference, because at the end of the day, if you're going to allow a little bit of botrytis in the fruit, or if you're going to ferment the fruit with a different yeast, is there going to be consumer acceptance of these wines? So we set out to do a study comparing wines fermented with EC118. So there was zero botrytis infection in the fruit, and these were wines produced from grapes that were up at 27.5 Brix. We compared that to EC118 fermented wines that had the 10% botrytis infection. And then we also compared the S. bayanus fermented wines that had 0% botrytis infection. We carried out the consumer preference study at the CompuSense facility in Guelph. There were 153 consumers that evaluated the wines. Each participant received each wine one at a time, and they scored the wines on a 9-point scale. And 1 would be dislike extremely, and 9 would be like extremely. And we determined the preference from their liking score. Okay. Now, CompuSense has made it very clear to us that a score of 6 or above means that there's good consumer acceptance of the product. And also, just to put it into perspective, they told us that typically anything that gets over a score of 7 is considered excellent preference, but that's usually reserved for products like chocolate. Okay, so I just want to put things into perspective before we look at the data. So the data that resulted, if we first compared the Cerevisiae produced wines without botrytis compared to with botrytis, we see that the scores were almost identical for consumer preference. What it also means, though, is that the score was above 6. So that means good consumer acceptance of these wine products. When we also evaluated the wines made with the S. Saccharomyces bayanus, on paper, it had a slightly higher score, but it was not significantly different from the other scores. So I cannot say that the consumers preferred the wines made with Saccharomyces bayanus, but they certainly didn't dislike them more than the wines produced with the EC118. So we were very happy with the consumer preference study here. We've got good consumer acceptance of Appassimento wines, a little bit of botrytis, up to 10% infection that we tested is not negatively impacting the wines, and we feel we've got a strong contender in this new yeast to look at commercial application for this yeast. So my last slide is a summary slide. So ripening grapes off vine after harvest to produce Appassimento wines represents a new and exciting wine style for the Ontario industry. It can allow vintners to overcome some of those climatic barriers and develop a unique signature wine style for the region. The process produces full-bodied red wines of high quality with consumer appeal. I'd like to emphasize that because that's important for a new style of wine coming onto the market. And wine flavor can be moderated through the drying method that's chosen to dry the fruit, through the choice of the fermenting yeast, and through the level of botrytis infection in the fruit. Now, this was a very large project, and I do want to take a second to acknowledge all of the partners that contributed to this project. We had a large team from Vineland Research and Innovation Center, and I see Bernard and Kimberly here in the audience today. We had collaboration with Niagara College. We have had a whole slew of individuals here at CCOVI at Brock University. But I would especially like to thank first Jen Kelly, who's a PhD student that did all of the work on the yeast strain and carried out the consumer studies for the botrytis and the yeast wines. But also a special thank you to Fred DeProfio and Lisa Dowling, who were the two key individuals in charge of making all the wines. And because of the consistency between the replications of each treatment, we were able to have a very robust data set to be able to draw the conclusions that we've been able to do today. So thank you to both of you for a great job. We had a number of industry partners that were donating their facilities and the grapes to allow us to do these studies. The main program was funded through the Ontario Ministry of Research and Innovation through the ORFRE program, and we also had some contribution from an Ag Canada program in the first couple of years of the project. So with that, I'd like to thank you for your time, and I think we've got a couple minutes for questions.
A couple of questions. I've heard this year of a producer who is keen to harvest earlier, and I've heard as low as 18 Brix is target. Now, given that there is metabolic activity post-harvest in the berries, do you think there's a point at which it's too early?
I think there will be. Do we have scientific evidence to back that up? From our studies, we don't. We specifically chose to take ripe fruit and further concentrate it. And the reasoning behind that was if the fruit is extremely underripe, and Cab Franc at 18 Brix would be considered fairly underripe, what would be the impact on concentrating the green flavors and specifically compounds like methoxypyrazines? Will there be further degradation of the methoxypyrazines through the drying process? Methoxypyrazines get degraded because of sunlight and heat out in the vineyard. So in a protected environment, I'm just not sure that phenomenon occur to a level that would be desirable. And keeping in mind that you're concentrating the fruit through the drying process, you're going to further concentrate. You would be at risk of further concentrating those green characteristics. We would have to do the study to prove that, and we just didn't dry fruit that was that low. The lowest fruit we brought in the day for Jen's study, although we were targeting 22, 23 Brix, her batch of fruit came in at 21.5, and but certainly didn't have those green characteristics to them. I would be a bit hesitant at 18 degrees Brix, but I think certainly that would require a study for me to be able to show some data to support that.
Second question. When can I get my hands on the Brockus?
We've had a few questions about that. So we're just looking at this stage of how to go about the next commercialization steps for that yeast. It's a yeast on agar plates right now, so it's not in a freeze-dried form. So it takes quite a bit to build the culture up to be able to use in wine production. But now that we've got the consumer preference work, we're going to be in contact with yeast companies to look at some freeze-dried trials, hopefully, and then the next stages for some small-scale commercial experiments. So we'll keep you in mind when we need the small-scale commercial production.
Anybody else? Well, first of all, thank you, Debbie, very much for the work. Little gift.
Thank you.
I think you had something to do with as well. I just want to bring your attention to next week's lecture series, which is COVI's senior scientist in viticulture, Dr. Jim Wilworth. He'll be on time next week. Who will be presenting his work on understanding bird activity in Ontario vineyards and bird deterrent strategies that he's been working on as well, so really relevant to people around the world. So look forward to seeing you all next week, and thank you again, Debbie.